Hello everyone, welcome back to Trixie Wikipedia. After a long gap, I am here with Steroids Part 3. In this video, we will discuss about classification of sex hormones with its example and structure. So, let's start with classification of sex hormones. There are three types of sex hormones: estrogens, androgens, and gestrogens. Estrogens are divided into three types: human estrogen and its derivatives, stallion estrogen, and synthetic estrogen. When we are talking about estrogens, we have to consider the estrane nucleus. Estrane nucleus contains 3 6-member ring and 1 5-member ring. The 3 6-member ring known as perhydrophenanthrene nucleus, and this 5-member cyclopentane ring is fused with this perhydrophenanthrene nucleus. And estrane contains 18 carbon atoms. So, at 18th position, methyl group is attached to the basic skeleton of steroids. Next is androgens. Examples of androgens are testosterone and dihydrotestosterone. And when we are talking about androgens, we have to consider the androsten nucleus. Androsten nucleus contains 19 carbon atoms. So at 19th position, methyl group is attached and we will get the structure of androsten. Next is gestrogens. Examples are progesterone and nortestosterone. When we are talking about gestrogens, we have to consider the pregnant nucleus, and pregnant contains 21 carbon atoms. Basic skeleton is the same as that of androsten. Only at 17th position we have to attach ethyl group and we will get the structure of pregnane. In this video we will discuss about estrogens in detail. We have already discussed about androgens and gestrogens in previous video, steroids part 1 and part 2. So let's start with estrogens. There are three types of estrogens: estrogen derivatives, stallion estrogen, and synthetic or non-steroidal estrogen. Examples of estrogen derivatives are estrone, ethinyl estradiol, and estriol. Examples of stallion estrogens are quinine sodium sulfate, and quinine. And examples of synthetic or non-steroidal estrogens are diethylstilbestrol, chlorotrianisine, dienestrol, benzestrol, and transstilbene. We will see the structure of all these examples one by one. These are the structures of estrone, ethinyl estradiol, and estriol. If you analyze all these structures, you will find these structures contain estrane nucleus, 3 6-member ring and 1 5-member ring fused with each other, and at 18th position methyl group is attached in all the structure of estrogen derivatives. We will see the structure of estrone first. In estrone, on is a suffix, so in this structure ketonic function is present. A ring, this first ring, is a completely unsaturated ring, or you can consider this ring is a benzene ring. In all the structure of estrogen derivatives, you will find the A ring is a benzene ring. Next is position number 3. At the third position, hydroxyl group is present in all the structures. So, in estrone, at the third position, hydroxyl group is present. At the 17th position, this ketonic group is present. This is the structure of estrone. Moving towards the structure of 17-beta estradiol. This diol indicates 2 hydroxyl group is present in this structure, and this 17 indicates the position and beta indicates the type of bond. So, in estradiol, 2 hydroxyl group is present. First hydroxyl group is present at third position, and second hydroxyl group is present at 17th position. and this group is attached by a beta bond. And basic structure is the same as that of estrone, and a ring is completely unsaturated ring. Next is ethinyl estradiol. This ethinyl indicates ethinyl group is present in the structure of estradiol. Means basic skeleton of ethinyl estradiol is the same as that of estradiol. Only this ethinyl group is attached at the 17th position by alpha bond, and we will get the structure of ethinyl estradiol. Next is estriol. This triol indicates 3-hydroxyl group is present in the structure. Basic skeleton is the same as that of estrogen derivatives, that is A ring is a completely unsaturated ring, B and C rings saturated rings, and D ring is a cyclopentane ring. At the 18th position methyl group is attached. Now triol indicates 3-hydroxyl group. First hydroxyl group is attached at the 3rd position. Second hydroxyl group is attached at the 17th position, same as that of estradiol, and third hydroxyl group is attached at the 16th position by alpha bond. Next is stallion estrogen. Stallion estrogens examples are quinine, quinine sodium sulfate, and quilanine. We will see the structure of these examples. If you analyze both the structures, you will find the basic skeleton is same as that of estrone. The structure of estrone is 3 6-member ring and 1 5-member ring. First ring is a benzene ring. At the 3rd position hydroxyl group is present. At the 17th position ketonic function is present, and at the 18th position methyl group is attached. This is the structure of estrone. And if you analyze the structure of quinine and quilanine, you will find the basic skeleton is the same as that of estrone. Only the difference is in quinine, unsaturation in between carbon number 7 and 8 in a B ring, and remaining structure is the same as that of estrone. At 3rd position hydroxyl group, at 18th position methyl, at the 17th position ketonic function, and unsaturation in between carbon number 7 and 8. Next is quinine sodium sulfate. The sodium sulfate indicates the salt form of quinine. In quinine sodium sulfate, we have to attach this sodium sulfate group at 3rd position by replacing the hydrogen from hydroxyl group, and we will get the structure of quinine sodium sulfate. Next is quilanine. The basic structure of quilanine is also same as that of estrone. Only the difference is unsaturation in between carbon number 6 and 7 and carbon number 8 and 9 in a B ring, and we will get the structure of quilanine. Remaining structure same as that of estrone only. That at the 3rd position hydroxyl group is present. First ring is an unsaturated ring. At the 17th position this ketonic group is present, and at the 18th position methyl group is present. This is about the stallion estrogens. Next is non-steroidal or synthetic estrogens. Examples are diethylstilbestrol, chlorotrianisine, and dianistrol. Name itself indicate these structures does not contain steroidal nucleus. So structure of diethylstilbestrol. The diethyl indicates 2-ethyl group is present. Stilbestrol indicates this bestrol indicates a benzene ring and hydroxyl group. You will find this benzene ring and the hydroxyl group. Or you can consider this ring as a phenol nucleus. This is the first phenol, and mirror image of this phenol is the second phenol. And both the phenol moieties are connected by carbon double bond carbon bridge. And first ethyl is attached to this carbon, and the second ethyl is attached to this carbon atom. This is the structure of diethylstilbestrol. Next is dienestrol. Dienestrol having the same structure as that of diethylstilbestrol. Only the difference is this group is attached by double bond and both the phenol moieties are connected with each other by carbon single bond carbon bridge, and this moiety is attached to this carbon by double bond, and this moiety is also attached by carbon double bond carbon bond. Next is chlorotrianisine. Chloro indicates chlorine group is present and trianisine indicates three anisine moieties are present in this structure. You can see three anisine moieties are attached with each other by this carbon double bond carbon bridge. First, second and third anisine moiety attached by carbon double bond carbon bridge, and this chlorine is attached to this carbon atom. This is all about the estrogens. Here estrogen is over. You can suggest the topic as per your requirement in comment box. Hope you have enjoyed this session and might be thinking something like this.